So my name is Sharon Aviran, and it's a pleasure to have Professor Sean Eddy here after several attempts that were canceled due to unforeseen events. Dr. Eddy is the Elmer Patterson Professor of Molecular and Cellular Biology and of Applied Math at Harvard. And he also currently serves as the chair of the Molecular and Cellular Biology Department. He's also an investigator at the Howard Hughes Medical Institute. His research program seeks to decipher the evolutionary history of life by comparative genomic analysis, and his lab develops computational methods for RNA, protein, and genome sequence analysis. He's the author of several widely used computational tools, including the Hammer and Inferno software packages, which I'm sure many people in the audience have worked with or encountered in their career. He's also a co-author of a very highly cited and widely used book on biological sequence analysis that many of us have multiple copies of. And most recently, his lab has started to tackle the question of how small genomes managed to specify biological complexity. Dr. Eddy received the Benjamin Franklin Award for contributions to open access in the life sciences. He's also a fellow of the American Association for Advancement of Sciences and the International Society for Computational Biology. So thank you for taking the time to come over here from Boston and visit and give the talk. Thank you.
Thank you, Sharon. It's great to be here. We have tried to do this for 3 years in a row, and it finally worked. This is not the worst example of this for me. I had a talk that had to be canceled a couple of times in a row in Vancouver, and they became convinced that I was trying to avoid them. And I had to do a layover in Chicago and almost missed the flight in Chicago. I actually got the pilot in Chicago to open the doors back up, which they will not normally do. I explained, like, I have to get to Vancouver. They will kill me if I don't. And they actually opened the thing back up, and I got on the plane. All right, so I didn't have to do that to get to here. I'm going to tell you about a body of work over many years. Picking up feedback, and I'm just wondering if this is off. It's off. About using sequence homology searches, about building the tools that we use for doing systematic homology searches and how they're getting better and better and how we're still not done. This is one of the first examples that really made an impression on the biological research community. This is a sequence alignment that was produced in the early 1990s or 1980s by Russ Doolittle using data that came out of Lee Hood's lab at Caltech. The sequence that Hood's lab had produced was done by protein sequencing, and it was the sequence of a normal human growth factor, PDGF2. And the way that I remember which one is which is all the question marks, because this came from Edmund degradation sequencing, which was pretty error-prone at the time. And then sitting in the database that Russ had accumulated was the sequence of a viral oncogene, P28 cis. Russ was working with his two sons. The older son knew how to write alignment code, and so the sequence alignment code was the older son's. The younger son knew how to type and was systematically accumulating, I think the kid was about 12 years old at the time, everybody's figure 1 that was showing a sequence, a protein sequence. The son was typing into the database that Russ was accumulating, which was the biggest protein sequence database of the time, a database called NUI. And what they were doing was, in every figure 1, comparing every sequence that was coming out to every sequence that they had previously seen. And there was a line printer, Russ told me this story, there was a line printer in the office, and usually it would stay quiet, but if there was an alignment, they would hear the chatter and they would know they got a hit. Now these days are completely different, as I'll show you. We get 100,000 hits to everything we throw against the database, but in these days, one hit was good news. This was a particularly good day because what this meant was that cancer is the derangement of normal growth control. We take that for granted now, but this was a major result at the time to see that a normal human growth factor gene was clearly highly similar to a viral oncogene. Therefore, you go immediately from that to going, okay, now I know something really important about cancer. So the world suddenly changed in the 1980s as sequences started to accumulate and people started doing sequence similarity comparisons from biologists saying you're never going to get anything from math and computation in biology, it's all experiment, to, oh my God, there's a really cheap way to get clues about the function of my protein. So we've completely industrialized this, of course, and as time has gone on, as I'll continue to show you, we've gone from simple, like pairwise sequence alignment to basically parsing protein sequences like they're not languages, but like they're speech or language into their parts, in this case, protein domains. This is a great example of the human dicer domain structure. In the database called PFAM, there is a model for each one of these. This acts more or less like speech recognition. You talk into your phone, it digitizes what you say into an alphabet, ships it up into the cloud, and compares it to statistical models that are pre-built of parts of speech. There's about 200,000 in typical speech recognizers, and then comes back with a parse. We now do the same thing with databases like Interpro and PFAM, standing libraries of about 20,000 models trained on hundreds of thousands of sequences in many cases that then walk down an incoming protein sequence and parse it into domains. So the technology has tracked historically speech recognition and continues to get better and better as both fields move forward. And that is shown by an example benchmark here, where the benchmark is not to try to convince you that anybody's method is better than anybody else's, but to convince you that as just over time, the whole field has moved forward. So what's being done here is to concoct a database where we have pairs of sequences that, because they have similar structures, and because people have looked at the structures, and there's a database called SCOP, where human curators have asserted that these similar structures are probably homologous. I'm being careful about how I say that because homology, evolutionary relationship, is something that we actually don't have ground truth on without a time machine. So these are asserted homologies based on structural comparison, and you can come up with binned true positives at different levels of sequence identity between the test query and a target. So you're basically saying at more and more difficulty, how many pairs, what fraction of pairs in that bin are recognized by different methods. So at the time that Russ was doing these experiments in the early 80s with his two sons, sequences had to be really identical. You can barely recognize things with sequence alignment at a +1, -1 scoring system, where you're just basically counting identities. But the moment we had statistical scoring, you got to a place where early versions of sequence comparison could recognize sequences about 35% identity. BLAST with improved statistics, actually, although you can still read papers that say that Smith-Waterman is more sensitive than BLAST, empirically it's not true, though it should be, and BLAST is better at about 25%. And then that got locked into people's sort of zeitgeist. If you ask people about the twilight zone of sequence comparison, where does sequence sort of crap out, they will typically tell you, oh, at about 25% protein sequence identity, that's what, below that, you can't see them, you're going to need structures. But that's not true anymore. I'll show you examples of the technology that we use now in SciBlast and profile hidden Markov models, exemplified by my stuff, where we can get down very typically to about 10% sequence identity. And we're not doing that by direct comparison between two single sequences at 10% sequence identity. We're taking the query and finding the homologs of that, building up a profile, and then using the profile to find the homology. So the field's gotten better and better and better, and part of the game is to push this as far as we can, because the better we can push the technology, the further back in evolutionary time we can see. I would like to be able to see the very earliest protein splits, divergences, going back to the last universal common ancestor of life. We can do that for some highly conserved proteins, but not for all. One important question is this a completely solved problem? At what point are you done? We don't know at what point we're done. This is, again, sort of like building telescopes to see back deeper and deeper in time. But here's an example of recent work from Kara Weissman in my lab, where it turns out that there's a so let me sort of walk you through this. The idea is there are many orphan genes or lineage-specific genes out there where you find, here's a gene family that's only in Drosophila. Many people would like to take that and say, oh, that's because there was a de novo gene origination event at the base of the Drosophilids. And so if I find orphan genes or lineage-specific genes and get lots of them, I'm learning things about de novo origination. Genes come out of random sequence at some rate. This almost certainly happens, and there's an interesting question of how often and when did it happen over evolutionary time. But there's another stupid hypothesis to explain that, which is that the gene actually does have homologs outside Drosophilids, and you just lack the power to find those homologs. It's a rapidly evolving, or it's a small gene, and if you had a better method, you would have found the homologs. And many of the papers working on de novo gene origination assume that the methods are perfect, and me, as one of the developers of the methods, I know that they're not perfect. And so it turns out that what you can do is you can take the BLAST bit score, or the Hammer log odds bit score, and plot it against evolutionary distance, here shown with examples from the Saccharomyces, sensu stricto, and other yeasts, where you can find examples where, as you compare a Cerevisiae sequence to itself versus its close relative versus next close relatives, and then it disappears and you can't find it further out. You can find examples like this where the score decay sort of goes below the level of detection anyway. This is a rapidly evolving short gene. You can't find it in Castelli or Cluveromyces or Aspergillus, but that you didn't expect to. You can tell from the exponential decay of the similarity scores themselves that you're going to lose power. Whereas there's other genes that are still way above detection limit, and they just mysteriously disappear in a plot like this. So Kara was able to show that almost all genes behave very simply, approximately following predictable decay curves, and therefore you could divide genes into two sets, ones where it's actually surprising that you lost them, their homologs, and ones where it's not surprising. So these are examples where this is just consistent with homology search failure. These are candidates for de novo origin. These are very interesting to evolutionary biology. But these are ones where the homology search has failed and where we could gain more power. There's many more of the homology search failure cases than there are things that I would think are interesting from a de novo origin perspective. And so those are the kind of examples that tell me that we still have lots of work to do on being able to look back in evolutionary time. So how are we going to do that? Where are we going to get the signal that lets us look back in time? Russ Doolittle, one of my heroes, wrote this article in 1981 that sort of laid out the future of the field, both for the power of genome sequencing, comparative analysis, and for where the methods were going to go. Russ predicted, as other people did at the time too, we're going to have the sequence of everything. And by comparing the sequence of all species, we're going to learn a ton about molecular evolution of life. But the problem is going to be that the computer methods that we use to do those comparisons are going to crap out, so we're going to have to make them as powerful as possible. And he foresaw several ways that you might do this, and some of them have played out, some of them have not played out. One of them has played out really powerfully. One thing about Russ, he was a biochemist, he didn't believe in statistics. He said maybe the statisticians will come up with statistical scores instead of using +1, -1 for sequence identity, but I don't believe in that stuff. That's not going to work. Well, that worked. I'll show you the Blossom 62 matrix that the Hennekoffs almost immediately introduced that made things better. He proposed an idea that we still don't use, but is a good idea. He said the business of finding homologs is an evolutionary question, which the relevant variable is the amount of branch length between sequence A and sequence B on an evolutionary tree going back to their common ancestor. The bigger the branch length, the harder the problem. If I have multiple homologs of A and multiple homologs of B, I should be able to reconstruct the ancestor of A, reconstruct the ancestor of B, and then compare the inferred ancestors. That's a shorter branch length. I should be able to merge evolutionary models with sequence comparison models and do better. That merger, although there's maximum likelihood phylogenetic models for inferring trees, and we use probability models of sequence comparison. Those are two fields that basically don't talk to each other. And my wife, Elena Rivas, other people in the field have worked very hard on trying to achieve a unification of those two methods, and it still hasn't happened for reasons I could tell you a ton about. Russ said maybe we should be comparing DNA sequences. Now the proteins have a bigger alphabet, you have more signal in them in principle, but you could be comparing codons to codons. That's 64 by 64. There's information in codon usage tables. There's information in gene structure. There's information that humans will use, biologists will use when they use synteny. There'll be a conserved gene and a conserved gene, and your rapidly evolving gene will be landlocked in the middle of that. You'll use synthetic gene order relationships to help you infer similarity. We couldn't do that when databases were oriented around just bags of protein sequences, but now that we have genome assemblies, and we almost have reliable annotations of those genome assemblies, we can start thinking about using gene order as part of the signal in an automated rather than manual way. He said, and now we have AlphaFold, he said, you know, maybe we won't have to compare sequences at all. Maybe we'll just predict three-dimensional structures, and then we'll compare the structures, and we'll be done. And we're almost there. And then the thing he said that was immediately something that we could operate on is use multiple sequence comparisons instead of pairwise. So first, just to sort of set up the multiple sequence and the profiles, let me sort of walk you through just a little bit of mathematical intuition for what we're going to do. This is the Blossom 62 score system. It's a bunch of integers that tell you when you align an A through a V to an A through a V pairwise. So regardless of where that amino acid I and amino acid J are in the protein, they're going to get the same score in a position-independent way. If they're an identity, and you can look down the diagonal, I stopped using this because it's not working, so I'm going to try to point. That down the diagonal, you notice that like an A to an A is getting a +4, a W to a W is getting a +11. So identities are not all the same. So where's that coming from? You'll notice that conservative changes are getting positive scores off the diagonal. There's some +1s and +2s you see. Some of the relationships that are on here, like look at E to K getting a, oh, you're a lifesaver, thank you so much. Look at the E to K getting a +1. So that's an acidic to a, you would think that's a very non-conservative change. So many people, when they get exposed to these scoring systems for the first time, are like, that's not right. That doesn't make any sense. Why are you doing that? There must, where did did the Hennicoffs make a mistake? There's a really important principle at the heart of what we do here, which is to operate with probability models where there's two models. There is a probability that whatever your, maybe it's a single query sequence or it's a profile, there's a probability distribution for what we expect the homologs of that single sequence or profile to look like. And there's a probability distribution for what we expect sequences to look like if they're just random and non-homologous. This will usually be a really simple and dumb model, like the product of the individual amino acid frequencies. This can be a hidden Markov model, or as I'll show you, a stochastic context-free grammar, or something that's a probability model of sequence and maybe even sequence evolution. So under really extreme assumptions where I say, okay, there's no insertions and deletions, it's local alignment, I just have a bunch of aligned residues to residues, they're all independent of each other, so all I have to do is think about them one aligned pair at a time. After I've made all these independence assumptions, which are completely unrealistic about how we think proteins evolve, then those probabilities I just show you factorize into something where you can end up at an additive scoring scheme. This is the Blossom matrix for an amino acid pair A and B that you just calculate by saying, okay, I know the frequency of A in random sequences, I know the frequency of B in random sequences, and I need some database of alignments that I trust that I can count how often do I see A and B in alignments that are homologous. If A and B show up more frequently than you expected them by their independent probabilities, then this ratio is greater than 1. The log is a positive score. You can scale it to anything you want to make it into an integer. If this is less, then this is a ratio less than 1. You get a negative score. It scales to a negative number. So this is so once you see this equation, and people did, They're like, well, I don't like what the Hennekoffs used for their alignment database. I don't trust it. Or I don't like what they used for their random sequence model. I'm working with transmembrane proteins, which have a different composition. So there's a cottage industry in making your own substitution matrix that's specialized to what you think your target frequencies are and what you think your background frequencies are. And you can make any matrix you want. And that's why when you do BLAST, there's a whole bunch of different score matrices that you can choose that are non-default. When you look at a multiple sequence alignment, this is one that almost fits on a slide, at least horizontally. This is a zinc finger, a C2H2 zinc finger family. You quickly see, all of us are familiar with the idea that not all cysteines to cysteines, not all pairs are equivalent. There's conserved positions and there's not conserved positions. So here it's a C2H2 zinc finger alignment. So there's a C, there's a C. That cysteine is pretty much always there. That cysteine is pretty much always there with occasional exceptions. I'm looking for the histidines. There's a histidine, which should be almost completely conserved. There's a histidine, almost completely conserved. There's other residues that show you strong conservation, residues that don't. There's insertions relative to consensus that are allowed in certain places, and this all makes sense if I showed you the three-dimensional structure of a zinc finger where the insertions and deletions are allowed. All that information shows up in a pretty accessible-ish way by looking at a multiple sequence alignment of the family. You look at this and you say, you know, what I really want is not this 20 by 20 very position-independent matrix. I want to have a scoring system that says there's 20 scores that I assign to the 20 amino acids in position 6 of my profile. And at position 7, based on what frequencies I see of amino acids occurring in that column, I'll make 20 scores for that. I want to have an insertion gap open, gap penalty, a gap open, gap extend penalty that is position specific too. So I need something on the order of 22 parameters that I'm going to set at each column of my multiple sequence alignment. That created a problem. Now you have to sort of understand what you're doing. You need to have some math that's additional for like, now your whole scoring system is going to come from one alignment because you're going to make a profile of it. You're going to be setting thousands of parameters from just this number of observations. And so you better have some guidance, especially for those insertions and deletions. People started to figure out that, okay, parts of this are solvable. The emission scores, the alignment scores for seeing a residue A at a particular position K, so this is just, instead of aligning A to B, residue to residue, this is the probability that I see A at a particular column. But the frequency of A stays the same sort of idea. It's still a log odd score. This is still an additive score that I would use for alignments. This was fine for ungapped sequence alignment. It turns the BLOSSOM matrices into a position-specific scoring scheme. And after a little bit of fighting in the literature, it was sort of clear to everybody by the sort of late 1980s that this was the right thing to do. And it is the right thing to do. It's derivable from Bayesian inference. But nobody knew how to do the insertions and deletions. And so the next step in the field was a really important preprint that came out from the Hausler lab, done by a postdoc at the time, Anders Krogh. This was a group that knew about speech recognition. Earlier algorithms had already been arbitraged into computational biology. What were called dynamic time warping algorithms in the 60s became sequence alignment to us. And then hidden Markov models, which were the state of the art in speech recognition in the 1980s, became state of the art in computational biology from this paper, which was a really pioneering piece of work where the idea for the emissions is the same as what I just showed you. There are a series of consensus states that correspond to columns in the alignment, and they have probabilities for the 20 amino acids at each one of the consensus positions. But you also have delete states and insert states. And these arrows are state transition probabilities that say there's some probability of like going into an insertion and some probability of continuing the insertion. That's a gap open probability and a gap extend probability. And then the insert state generates random amino acids sort of at the background if you want, and then comes back in. So now the insertion and deletion, the gap scores were not arbitrary. These are probabilities. They have to sum to 1. If you try to make it very cheap to insert a gap, you're making it very expensive to not insert a gap because of the need for probabilities to normalize. And that meant there's a natural optimum. The natural optimum for BLAST is to set the gap scores to 0. You'll get really bad alignments. but they'll be the highest scoring alignments. If you try to do maximum score, maximum likelihood with these models, you actually get a biologically sensible answer because everything is sort of balanced against each other, which again makes sense from a statistical inference perspective and was one of the real strengths of these models, that it put it on a footing where we knew what we were doing from a probabilistic inference perspective. So we have a homology model that is a profile hidden Markov model. We have a non-homology model that's actually still really stupid, and I could talk about that as we try to replace the denominator of what we do with these models, because sequences are not random IID. They are more complicated than that with a lot of biases. But this sort of framework of what we do with these models, which sort of stands in sharp contrast to where speech recognition is right now with discriminative deep learning models, this style of generative probability models actually gives us a lot of power. We make complicated models by drawing them as graphs. We draw what we mean by the biology, and everything else sort of follows automatically. Because they're probabilities, we can sample sequences from the model, and if they don't look like the biological sequences, we know the model's wrong. We go back and change the model. These are Bayesian models, so we can incorporate prior information into them, and they explicitly handle uncertainty, including alignment uncertainty. There's nothing in here about, given an alignment between the two, we actually run what are called alignment ensemble algorithms that sum over all the possible alignments. So as the sequences get further apart, alignments become uncertain. BLAST has to choose one, but it knows it's going to be wrong for distant sequences. The HMM algorithms are on a good footing so that we sum over all the ensemble of all possible alignments, which can be done in the same time it takes BLAST to find its optimal one. And that gives us power for more distant comparisons too. So as an example of sort of drawing the models that we want, the model that I showed you earlier from Anders was a model of a complete protein domain, but what we want to be able to do is identify protein domains in full-length protein sequences. That domain might occur more than once. It might be a local alignment. It might be a fragment of a domain. And there's other sequence in there that's not homologous to the domain. So we just draw that as a graph. The Hammer model currently in the software is that there's non-homologous sequence at the beginning of the sequence. That's part of the model. There is a copy of the domain. There is non-homologous sequence following the domain. There might be more than one domain, so it's allowed to cycle around. There might be residues in between the two copies of the domain. You're allowed to start anywhere in the model. You're allowed to end anywhere in the model. So this is local sequence alignment, but it's implemented in the model, not in the algorithm. It's implemented in the choosing how you draw the model, saying these are the allowed transitions in the model, and this is how I'm going to parameterize them. So this gives us all of this is generative probability modeling that sort of we understand exactly what the model is trying to do, and the rest of it is automatic implementation. Okay. That is how I learned how to do probability modeling. The problem that I was really interested in is I'm an RNA biologist. So I wanted at the time to be able to find RNAs by sequence, by structure alignment. The thing about RNAs is that we as biologists do not draw them as lines. We do not draw them as little boxes of domains with these little things. We draw them as two-dimensional pictures as secondary structures. Now, of course, whether it's a protein or an RNA, ultimately, a functional RNA, ultimately it's folding up into an interesting tertiary structure. Proteins, there's not too much of a useful middle ground between the sequence and the 3D structure. There's, of course, helices and sheets, but the helices and sheets do not constrain the sequence well enough that it's useful for sequence comparison. You just have to, it's a two-state physical thing. Either you do one or the other. So either solve the structure by AlphaFold or compare the sequences to reasonable approximation. But for RNA, what we do as humans, as manual RNA biologists, is we look at these secondary structures, and the Watson-Crick base pairing of a secondary structure, as I'm about to show you, is highly conserved. That secondary structure folds up, and I want to introduce a concept of what we in the RNA community call a 2.5D structure that's sort of in between. If you look, for example, at P1 and P4, these are two helices, P1 and P4 in the flat picture here. Notice no gap in between those two residues. What's happening is this and this are folding into a contiguous stack. So that base pair and that base pair are stacked on top of each other, and it looks like a continuous double helix. Same thing here, if I get this right, this is P3 and P2, no gap. That GC stacked on this CG, and you'll see in a second why I'm belaboring this. So when we draw them as 2.5D, we're trying to represent those stacks. Once you've seen those stacks and inferred them correctly, you are very close to the three-dimensional structure of RNA. An important point here is way before AlphaFold, humans were solving RNA three-dimensional structures at pretty high accuracy because the information in this sequence caused by evolution is extreme relative to what we get from proteins. There's also additional interactions. I want to point it out an additional kind here. This interaction of this GUCC, which now I'm going to block on where exactly it is. You'll be able to find it faster than me. The P2B loop, this CUGG, is pairing to the P1, P4 junction, which I think is sitting down here. That interaction is not a stem loop interaction, but it is a contiguous stack of residues. We call that a pseudoknot. Those are super important for understanding the three-dimensional structure. You can't get this three-dimensional structure right unless you understand that the pseudoknot is there, but we are unable to draw it in a two-dimensional planar picture, and the algorithms used for RNA folding typically ignore pseudoknots as well. It's only a few bases, so it's not much information, but absolutely critical for understanding the 3D structure. All right, so here's the magic. This is the sequence signal that we get from comparative genomics for RNA. This is a condensed version of the alignment of that thing I just showed you, which is a little RNA structure called a riboswitch. It's called a SAM riboswitch, I guess I should have said. Sitting in the mouth of this is a S-adenosyl methionine. This is an RNA that acts as a cis-regulatory molecule that when it binds SAM, it's in one conformation. When it's unbound to SAM, it's in a different conformation. And that's used as a regulatory switch in post-transcriptional control of messages that carry this in their upstream, in their 5' UTR. And when you align that sequence to lots of its homologs, what you get is a sequence alignment where in green, what I'm showing you, and let me just pick an example to make the point, this is a base pair from this column to this column. It's a G to C, but there's an A, and when that's an A, that's a U. When that's an A, that's a U. The next base pair over, usually a GC again. When that's an A, that's a U. So all the green guys are showing you pairwise covariation. It's maintaining the base pair. Evolution is making a change, but then making the compensatory change in such a stereotyped way that once you've seen an alignment with all of this green, you can absolutely tell where the pairs are. That's true also for the pseudoknots, where you can see the number of green guys in there. It's not perfect. The red guys are showing you where evolution is tolerating a little slop in there. The blue guys are showing you half swaps to GUs that are consistent with the structure, but not, because it's not a double change, it's not the kind of thing that you would absolutely rely on as compensatory substitution evidence for a base pair. This signal is really, really strong in RNA, and this is something that from the very early days of tRNA sequencing and ribosomal RNA sequencing, people were solving secondary structures, inferring secondary structures from this information. In the field, people do not use, I'll sort of make a strong statement for the sake of making the point. You've heard of M-fold. You know that there's programs out there for, like, give your sequence to it. It'll give you a thermodynamic minimal energy structure. That can be useful. That can be a guide, but nobody relies on it to be the true structure. Those are not accurate programs. But this, given a sequence alignment of the right variation that's accurate, that's been lovingly looked at by humans, produces accurate near 3D RNA structures. When the ribosome was finally solved, 98-point-something% of its base pairs had already been predicted by Robin Gutel. You can run very simple statistics on pairwise correlation. This is a statistic called mutual information, where for the alignment of the SAM riboswitch against itself, in the lower triangle, you're seeing the true base pairs in that structure, in the non-pseudoknotted structure, and in the upper right, you're seeing the mutual information calculation as a heat map, where you're seeing the P2, P3, P4, P1, and the pseudoknots standing out. Pretty much every base pair supported by comparative analysis. Now, this particular plot is produced by a piece of software called Artscape that's by my wife, Elena Rivas. And one of the things that Artscape solves is not just the ability to solve to infer where there are pairs supported by compensatory information, but also to do an important statistical test for when that amount of covariation is more than you would have expected by chance. And that's become an important question that if I know that I'm dealing with a SAM riboswitch, I know there's a conserved structure, and the question is, what is the best structure for this thing? But there's another question which arises from long non-coding RNAs, where is there a conserved structure at all? The link RNA could be transcriptional noise. The link RNA could be an undiscovered coding region. You don't know that there's a conserved structure there. You can always find one. You can give it to MFold. It'll show you something that looks like a great RNA structure. But you want to know, is the evidence good enough, better than I expected by chance? And that's a very important problem that RScape solves that I'm not going to go into. What RScape also does is has greatly improved the ability of people to infer even the details of 2.5D structures that make a big difference. So what I'm showing you here is an example of Elena's program where this is the SAM riboswitch again, which makes a great example for all the things I'm trying to tell you right now. The green are where RScape says there is statistically significant statistical support for the base pair in the sequence alignment from Rob Beatty's lab at Colorado. The structure that I'm showing you on the left is the structure that's annotated in the RFAM RNA structure database, or at least was at the time. In green are additional pairs that once you get to the structure predicted by RScape, so these pairs are proposed by RFAM and they are supported by the data, but there are additional pairs that are also supported by the data that RScape finds. And when you make the fold that has them, one of the things that happens, and this is why I was belaboring the point, look at this AU and this CG. To get the stack between P1 and P4, you have to be able to predict what's essentially looks like a lone pair. Lone pairs in RNAs don't happen. You don't get a single base pair. It has to stack on something to be stable. What it's stacking with is actually P1, but it's hard to see that in a 2D thing. But there's statistical support for the AU pair. So Elena's program actually predicts the lone pair, and then a human or anything that's been taught about coaxial stacking immediately recognizes that that's probably a coax stack. Same thing happens here. This gets predicted down to the next two base pairs, and now it becomes really easy to recognize the P2, P3 stack, which is supported right at its base by covariations going the two directions. So from the RFAM structure, you can't really get to the 2.5D, that's not to the 3D, but from the RScape structure, you can get to the 2.5D. And so that, getting those last details right was actually a big step forward for structure annotation of RNAs. Now, Elena works on detecting new RNAs and solving their structures. I'm interested in being able to find RNAs by structure comparison. And here the problem was, profile HMMs, I talked about the independence assumptions that were made by pairwise sequence alignment. Profile HMMs make some of the same assumptions, and one of the most notable ones is that individual positions are statistically independent of each other. Base pairs are distinctly not statistically independent of each other. You have to, once you see it's GC, AU, UA, the two positions are evolving in concert, and they can be far apart. They can be in different sides of the molecule. So profile hidden Markov models can't solve that problem. Profile hidden Markov models that we use for protein are linear models, one column at a time. What you need to do for RNA is move all of that math to trees, binary trees, that now the states of the model, instead of generating one position at a time, the state of the model has a 4x4 matrix of generating the guy on the left and the guy on the right as part of the same statistic. So there's a joint probability for the two residues. So this state can learn that there's a Watson-Crick base pair that can be almost anything. If it happens to be a highly conserved base pair, like this is a GC almost always, that can be what you emit with the 16 probabilities. It turns out that all of the algorithms that we use for sequence alignment can be then moved to alignment of sequences to trees. And it turns out, though I didn't know this at the time, that that was an existing model in the linguistics literature called a stochastic context-free grammar, an SCFG. So when we profile them on multiple sequence alignments for biology, we call them profile SCFGs. They had never really found a use in linguistics. There aren't many languages that show nested pairwise correlation. The big constraint of a binary tree is that pairwise correlations have to be disjoint, one base, you can only base pair with one partner, and they have to be nested. Proteins don't give us that. It's sort of an all versus all mesh, so is language, and so some of you might know from deep learning stuff that the modern models that people are using, called transformers, are looking at pairwise correlation structure, but in a sort of all versus all way. This is a very restricted way of looking at it that fits our RNA structure almost perfectly, but gives up pseudoknots, because the pseudoknots would be an example where these interactions would be crossing, and they can't be captured by the binary tree. The algorithms for working with these look like sort of a unification of sequence alignment with RNA folding algorithms, and they share properties with how algorithms for RNA folding work. And the basic idea of this, without going deep into it, is that here's a tRNA as an example. The structure of the consensus model for the tRNA looks like a binary tree instead of a line. And then when you get a result of aligning a sequence to this tree, so you sort of wrap the sequence around what's called the yield of the tree, the edges of the sort of edge of the tree, you end up with a result that looks like a sequence alignment, but it's really an alignment to the tree, and there's an extra line that's showing you that. So this code is showing you this is the open parenthesis corresponding to that closed parenthesis. This is a GC pair, and the sequence is conserved. We go to this guy. This is a CG mismatch. The little colon is special code in an RNA alignment saying, okay, I know it's a mismatch, but that guy is paired to that guy, which is a compensatory pair, GC. So that's still getting positive score, even though there's two mismatches in it. And that's coming because we're aligning at the RNA level. So it's important to know this is a principled mix of sequence and structure. The sequence is also in there. If that was a completely conserved GC, we would have 100% GC as the emission probabilities. If it's only Watson-Crick pairs, we'd have 25% for the 4 Watson-Cricks of the 16 possibilities. So we're capturing both sequence and secondary structure conservation. Okay. We had that model in 1994, and we couldn't use it for anything practical because I failed to tell you, if I'm aligning a line to a line with BLAST or with profile HMMs, I build a quadratic dynamic programming matrix. The time and memory that it takes me to do the alignment is proportional to the product of the length of the query and the target. I can accelerate that with techniques we use for BLAST and Hammer, but it's n squared. For this, I have one state in the model that now corresponds to 2 positions in the sequence. So my dynamic programming lattice is 3-dimensional. I'm at least n cubed in memory. And then for a technical reason, there's an additional step of how I put so-called bifurcations of 2 stems at once together that means they're n4 in time. So for a typical RNA of 100 to 1,000 nucleotides, the hit in time means I've taken a 10,000 to million-fold hit in time relative to sequence alignment. Sequence alignment is already a high-class problem. So unless you figured out a way to accelerate this, you're not going to be using it on whole genome searches anytime soon, but that was the goal. So this is the part where there's like the Rocky training montage where like then paper after paper after paper over a very long time from my lab found more and more techniques to drop the memory and speed up the algorithm. We use techniques that I learned how to do in the context of profile HMM searches, and by about the mid-2010s, we started to have a program we could actually use. And by the time we actually had a program that we could actually use, the entire lab, except for me, had forgotten why we had started building it in the first place. So Eric Naraki, who was working with me at the time on the code, the first time that we had a version that could run whole genome searches, took every RFAM model and asked, now that we can run a full-blown secondary structure-based search, which RFAM models gave us the most new hits in searching the whole sequence database, genome database at the time? And they were a lot of the usual suspects: spliceosomal RNAs, transfer RNAs, things that really conserve their structure but not a lot of their sequence. But the number one on the list are the catalytic group 1 introns. And this is what I was working on as a graduate student that motivated me to get into this field in the first place. I wanted to be able to find catalytic group 1 introns. So all of a sudden, we finally had the tool, and it turned out that many of the questions that had initially motivated me to start developing it were still open for a whole battery of reasons. So here's one, just as a sort of example of one of the things that we think we can do now. So you may know that in splicing, there are group 2 introns that are catalytic and they're relatively rare. And the spliceosome that we know and love in mammals is thought to be the evolutionary descendant of the group 2 introns. The spliceosomal RNAs are thought to be abstracted away from the big catalytic structure of the group 2s, and that people have now solved structures of the group 2 and the spliceosome catalytic core, and this is a pretty solid story. In archaea have introns, but they're of a special type called bulge helix bulge introns. And this paper from Tacchini Valentini proposed, and so group 1 introns have never been found in the archaea. One of the big mysteries about the group 1 introns is their weird phylogenetic distribution. They had never been seen in the archaea. And Tacchini Valentini said, this is going to be just like the group 2 to spliceosomal RNA story. Group 1 introns evolved into the bulge helix bulges, and then when extinct, the bulge helix bulges are like the Cheshire Cat smile left behind from the dead group 1s. But as you saw in the very beginning of the talk, an alternative hypothesis is just that nobody has the power to recognize group 1s. You wouldn't know one if you found one. You'd need to be able to see the secondary structure conservation, and people are just dumping genomes of archaea into the database at this point. If they were there, you wouldn't even know it. And so of course they're there, and the moment that we could do the search, we lit up a whole bunch of group 1 introns in the database. These are big, complicated structures. There are about 300 nucleotides of catalytic core. They have open reading frames in them that further complicate things because there's a whole protein coding gene sitting in the middle of the catalytic core, and the software deals with that just fine. So I was super happy with this paper. This was sort of like coming home or something. After 30 years of work, we did one of the searches that we wanted to do, and so this is circa 2018. So now we're sort of looking at what we can do in bacteriophage genomes with catalytic introns, because I'm originally an RNA biologist interested in phage biology. But in the meantime, what the lab has become is a computational technology lab. So what Sharon mentioned at the beginning of the talk is there's these 2 software packages from the lab, Hammer and Infernal. Hammer implements the profile HMM software that people are using actually to produce the alignments for AlphaFold. Google uses it, and to produce these diagrams that we often see in papers based on protein databases like PFAM and Interpro. And then Infernal doing the secondary structure code, a more complicated code base, a slower code base, but now finally practical, that the RFAM database rests on top of. And for that, all that database work, we rely very heavily on a collaboration with Alex Bateman, Rob Finn, and others at the European Bioinformatics Institute, a collaboration that's also been going for about 30 years now. So now we all know about AlphaFold, and so now we have a couple things that are really important. We have really deep sequence alignments produced by, for example, by Hammer and PFAM. So here's an example from one of the early sort of pre-AlphaFold papers showing, once you have 100,000 sequences, you start to see the subtle covariation pattern, which looks an awful lot like the contact map of a protein structure. And based on that pairwise signal, people in David Baker's lab and elsewhere started solving or inferring 3D structures, and of course now we all know that this works. Now it's actually a little unclear how AlphaFold is working. AlphaFold turns out not to need 100,000 sequences. It only needs about 30, and I've seen it work on 5. So it's not clear to me at all that it's relying on pairwise covariation information, but this is still part of the story that we think is part of what AlphaFold's information is using. So what my lab, because we're interested in homology search, not structure prediction, is now looking at, inspired by the fact that we have always tracked behind speech recognition. Speech recognition has completely been revolutionized by deep learning transformer models. We ought to be able to use these more complicated models that are closer to 3-dimensional contact information to replace profile HMMs with something more powerful. We have worked with Bayesian networks. We have worked with statistical models called Potts models. We're starting to use these transformer models. We think that we have a ton of power that's now enabled by the deep alignments. But the alignment problem is pretty serious, but we know how to deal with this. We've had a lot of experience of accelerating profile SAFG algorithms and making them practical. There are some other issues here that I can talk about that I think are going to bite us, but we're going to work through those. And we're going to adapt these things that are being used, like AlphaFold, for structure prediction Now our goal is to adapt them to homology search. So that's ongoing work. These are the people who so I actually still code, I promise. And so a lot of the work is still mine, though going slower and slower as time goes on. And then there's a bunch of great people in the lab, including Sam Petty, who just got a job, who works on AlphaFold, and our collaborators at EBI, Eric Naraki, who still collaborates with me at NCBI. I showed a little bit of the work from Carl Weissman, who's now at Princeton, and Gray Wilburn, who now is a data analyst for the Minnesota Twins. And I've worked with my wife, Elena Rivas, for many years, and she did the RScape work I showed. And with that, I'll stop, and I'd be happy to take questions.
Point. What is the practical application knowing that?
I don't think there might not be one, but that question reminds me of a story that I love when I get a question like this, which is there was when they were building the superconducting super collider, the physicists, they were all like trying to get the funding from Congress, and they had someone testifying in front of a physicist testifying in Congress, and a senator said, what is the practical use of the superconducting super collider? Will it help us defend the country? And the physicist said, it will make the country worth defending. And I think knowing about the origin of life is one of the deep questions of biology. And one of the striking things that sequence comparison is telling us that I find actually amazing is that the last universal common ancestor was really complicated. It was a bottleneck organism. And it was not the first thing that crawled out of the soup. It had ribosomes, it had RNA polymerase, it had DNA polymerase, it probably had at least 300 genes, possibly more, based on these ancestral sequence reconstructions that people can do. So there's a whole bunch of evolution between Luca, the last common ancestor, and whatever the origin of life looks like. And we can still get information about that because many of the genes in Luca are themselves paralogs, like the aminoacyl tRNA synthetases. Two big families of them, 10 and 10, they're homologs of each other. So we can see that branch length to the duplications of the aminoacyl tRNA synthetases. And those branches are deeper than the last common ancestor. So there is information available to us going back even deeper than Luca from sequence comparison. I'm just super fired up about how much information we can get. The other thing I'd love to get is a different organism than the eukarya, archaea, and bacteria, because if we could find a different lineage, we could potentially go back further in time. So is there a fourth domain that's even older on this planet or others? Could there be extant lineages that horizontally transferred their genes into the extant lineages, and maybe we can get back that way? And there is a lab at MIT that thinks that's true and is working on that. So I think we actually have a fair amount of traction on the very basic question of where the molecular origin of life was.
All right, before we go on to any other live questions, we do have one that came over on Zoom. So I will read it. Are there good approaches to examining patterns of sequence similarity that can flag likely cases of convergent evolution? Perhaps the rate of decay of similarity is different that for similarity due to homology.
This is an excellent and very thorny question. So we now believe, everybody in the field now believes, that the models we use are sufficiently powerful to see many cases of convergent rather than divergent evolution. If you were to pick, if you do as a thought experiment, make a bunch of random sequences and select the ones that function as globins, they would look like globins. They would be able to bind heme. They might actually be in some well-in-sequence space. Like if you imagine some big arbitrary sequence space, there is a place in that space that is the globin. There is a place in that space that is the kinases. And if you randomly sampled enough sequences, some of them would fall in that peak and they would be globins. And they would be convergently evolved because you randomly selected them. As opposed to you started here and you did a Markov chain Monte Carlo and you sort of walked around and evolved the globins. That's another way of sampling that peak, and that's divergent evolution. At the end of the day, it's sort of a numbers game of like, how big is that peak? How easy is it to throw a dart into sequence space and hit a peak? The more complicated the protein structure, the less likely we think it can be convergently evolved. So the cases we really believe are things that are really simple sequences, short period repeats that make up like the antifreeze proteins in fish blood in the Antarctic and the Arctic, which clearly separately evolved the same tripeptide repeat. And coiled coil domains, which are super popular in proteins, look like they've convergently evolved again and again. Then you get to things like TIM barrels, where the TIM barrels are a very simple repetitive structure, but they're big. And some people think, well, that can only be divergent versus convergent. But the active site of enzymes that are TIM barrel enzymes is all different places. And so that's sort of support for, well, maybe they're convergent. And you don't really have a super principled way of arguing that. But the questioner said one of the ways that people have used, which is to look for certain patterns, basically consistency with trees. There's a guy, Doug Theobald, who's written papers about this. Divergent evolution predicts that the sequence evolution is going to follow the tree that you expect given the other sequences in the species, whereas under convergence you might not get something that's congruent with the tree. And that should be true not just for selected positions, because they'll be forced in some sense to follow certain patterns, but it should also be true for unselected positions. So at least for sequence comparisons that are close enough that you can still see the neutral evolution and get information out of it, that it's not saturated yet, you do have a test that can distinguish convergent from divergent. But for the distantly related things, it's very tricky.
Any more questions?
To get back beyond where...
Right, so we often have like orphan genes, we don't know where they came from.
Yeah, so I mean, that's why we would like to have more powerful methods. One way that actually works pretty well in many cases that's manual and nobody's automated is to use synteny. So like when you're in Saccharomyces, for example, with a Saccharomyces orphan, Kara has shown and other people have shown that the genes flanking your orphan gene are often conserved. So you can find where they are in the other species. And then you look in between and you're like, oh yeah, there is a gene in between there. And then you look hard and it's like, okay, yeah, I actually believe that those are probably the homologs. So having conserved outposts flanking your gene does give you information, but there's only been a couple attempts that I know of for people to try to automate that. That's a very common trick for people to use when they're actually staring hard at their favorite sequence, but not an automated thing at this point.
Thinking about gene ordering and things like this, do you think we're ever going to get to the point as a field of thinking about models of genome evolution, like building generative models of genomes and trying to understand basically the space of possible operations on how genes can get moved around or duplicated and things like this?
Yeah, I do. I don't think that I know how to do it. Every time I've tried to think about it, I've gotten a little daunted by it. You then, very quickly as you go out to genome scale, as you know, I would then have to deal with inversions and duplications and transpositions and stuff like that that does not fit the dynamic programming model. So you're in Pavlov-Pevsner land or stuff where your algorithms are going to get fairly complicated, but there's all kinds of heuristics one could use that if you could write down the probability model and be able to score a proposed alignment, even if it's a heuristic alignment, you can still use generative models to deal with that. So I think that there is traction there. I would love to be able to do more work on it. I think there's a lot of information there. I mean, intron-exon structure is conserved. You get information about all kinds of evolutionary events that are happening at that kind of scale, and every event that you can take account of gives you signal in these kind of inferences.
Using these approaches to predict the structure of SARS-CoV-2, these approaches tell you anything about the evolutionary constraints on the virus?
So that's an example where we don't get a lot of information from the kind of stuff that I do, which is basically a timescale thing. Where we're really focused is on very remote comparisons. And so to me, the question about like SARS-CoV-2 would be a question about like coronaviruses or RNA viruses in general. But most of the interesting questions about the pandemic have to do with the population biology within SARS-CoV-2 populations. And those are much more short time period things where the alignment is obvious, relatively speaking, and then the inferences are sort of using a different, they're sort of different questions. So we haven't really done anything with that at the protein level for SARS-CoV-2. Now, the RNA structure of the virus is a different thing, and there's a lot, there are, I haven't looked at this myself, I understand there are functional RNA elements in SARS-CoV-2 that people have studied. There's a frame-shifting pseudonaut that's important for its biology. My lab hasn't looked too much at that.
Questions? Do we have any more online? No. All right. Let's thank the speaker.
Thank you.
Thank you very much.